I'm Dilrukshi, currently working in Wayamba University of Sri Lanka. Today I am going to present the soil improvement method based on plant-derived urease-induced calcium carbonate precipitation. Other authors of this study are Professor Kawasaki and Professor Nakashima from Hokkaido University. This is the overview of my presentation. Actually, the concept of biotechnology for the soil improvement has spread in the field of geotechnical engineering around in 2005. Since then, different biogrout formation mechanisms have been identified. However, the soil improvement using urease-induced calcium carbonate precipitation method is the most widely explored method. I will explain how calcium carbonate precipitate using urea and urease. This reaction is the urea hydrolysis reaction, and it results ammonium and carbonate. This reaction is catalyzed using urease enzyme. This enzyme may be from plant urease or maybe from microbial urease. Then when calcium ion present in the system, the carbonate react with the calcium ion and it will precipitate calcium carbonate. Hereafter, for the soil improvement using plant-derived urease-induced calcium carbonate precipitation method, I use PDY-CCP method, and microbial-induced calcium carbonate precipitation method, I use MICP method. When compared with MICP method, PDY-CCP method has several advantages. Therefore, I considered PDY-CCP method for my study. So my objective was to reinforce the sandy specimen to an estimated UCS ranging from several kilopascal to megapascal using watermelon seed urease-induced calcium carbonate precipitation technique, and also to identify the range of applicability of this method, such as to mitigate liquefaction, create artificial soft rock, and etc. Under the materials and equipment, I use the commercially available the poorly graded mica sand along with some other chemicals, and also I use the watermelon seed extract as the plant-derived urease source. This is the extraction procedure of the urease enzyme from the watermelon seed. First, we crush the seeds and soak about 1 hour with stearin. After that, we collected the filtrate. Then this is the collected filtrate. It consisted with fine particles. To remove the fine particles, we use the centrifuge. And this is the sample after centrifuging. Then we use both these filtrate for the urease activity testing because we wanted to check what happened to this filtrate when we do the centrifuging. Then we conducted the test tube experiment in order to check the rate of calcium carbonate precipitation with different combination of calcium chloride, urea, and urease. Actually, the purpose of doing this test tube experiment is to understand when we use this solution with this concentration for the sand solidification, what happened inside the sand specimen. To understand that one, we conducted this test tube experiment using polypropylene test tubes. Then for the syringe solidification test, we use this kind of a small syringe and we place sand into 3 equal layers. After that, we added the calcium chloride urease mixture. Then we allow to penetrate this solution through the sand specimen, and while allowing the penetration, we close the outlet valve. Then the new solution with same concentration of calcium chloride urease was added daily into this specimen. Then we allow to drain out the previously injected solution while adding a new solution to the specimen. Then we measure the calcium ion concentration and pH in the outlet solution once in every 2 days. Then we conducted this experiment in an incubator in order to maintain constant temperature. Then after solidifying the sample, we use needle penetrometer to measure the UCS of the solidified samples. Then by using these equations, we calculate the theoretical amount of precipitated calcium carbonate within the sand specimens. Then we will move to the results and discussion part. Then urease activity is increased with the increase of the concentration of watermelon seed extract. At the same time, the before centrifuge and after centrifuge solution, then urease activity is little bit decreased after centrifuging. And also the temperature also affect for the urease activity. For the maximum, the high urease activity was obtained around 50 degrees Celsius. According to the results of the test tube experiment, the rate of decrease of calcium ionic concentration is increased with the increase of the urease activity. When we come to the low concentration of calcium chloride urea, we can see the small amount of urease can be used to hydrolyze this small amount of urea. But for the 0.7 molar case, we need that small amount of urease is not enough for the precipitation of calcium carbonate. For the syringe solidification test, we conducted 2 control samples. One is from calcium chloride and urea, other without urease. The other control sample is using calcium chloride and urease without the urea. In addition to that, we prepared the several specimens by changing calcium chloride urease concentration. When we consider about the estimated UCS value variation with the calcium chloride urea concentration, the calcium chloride urea concentration increase from 0.3 molar to 0.7 molar, the estimated UCS is also increasing. We can explain this one clearly using these equations. This reaction, this urea hydrolysis, this 1 mole of urea react with 1 mole of calcium to form the 1 mole of calcium carbonate. When we increase the calcium chloride urea concentration, then amount of precipitated calcium carbonate also increase. Then that calcium carbonate precipitate within the pore spaces, then estimated UCS can be increased. The similar observation we observed for the the urease activity value. This is the variation of the estimated UCS with the urease activity. Here the 0.7 molar urease activity, 0.7 molar calcium chloride urea concentration, the estimated UCS is increasing with the increase of urease activity. But when we come to the low concentration of calcium chloride urea, when we increase the urease activity, estimated UCS not increase, it is decreasing. It is not like in 0.7 molar place. Therefore, the selecting the most appropriate urease concentration for each calcium chloride urea is important to important for the effective precipitation of calcium carbonate. We proved that the precipitate was calcium carbonate as calcium carbonate by using SEM images. Then we will move to the conclusion part. Then we obtained the estimated UCS of several kilopascal to megapascal by changing calcium chloride urea, urease activity, and curing time. And we obtained the estimated UCS after 14 days of curing time for 0.7 molar calcium chloride urea and 3.912 urease activity was around 3 megapascal. This is the maximum value we obtained from this study. By changing these parameters, there is a possibility to obtain a UCS value to mitigate the liquefaction and artificial soft rock formation. And also the change of this urease activity with temperature should be quantitatively evaluated in future to understand the effect of temperature on estimated UCS. And the urease obtained from the crude extract of watermelon seed has a potential to apply instead of the commercially available urease for the carbonate precipitation. However, the well-controlled distribution of calcium carbonate should be achieved to use this method from laboratory to large-scale field applications. These are my references, and thank you for your listening.